Connaissance,
recherche,
sciences,
dessin, mathématiques,
théorie,
physique, philosophie,
conférence,
archéologie, nature.
Jan Dausset is here forever among most of us, and in this second part of his life, within most of us also. He sometimes whispers, or may whisper, something like this: I intend to report, describe, and comment upon the festivals through the region of myself which I have called Spain. Certainly, his ties with Spain have been stronger than even himself realized in this first part of his life. He met a Spanish person who has been fundamental for his work and everyday life, Rosita, his wife, born in Madrid, and his continuous support. Spain is now the country which more transplants is performing in the world. This is in a great part due to the work of Dausset and a group of Spanish people close to Dausset. At the end of the 70s, a transplant law was issued in Spain, which was a copy of the French one. The French law had been inspired by Dausset, and most of citizens were in fact organ donors. Dausset had constructed the French HLA laboratory network. 4 laboratories in Spain were considered a part of this French network. One of these laboratories was directed by me, who had just come from London, from Hilliard Feinstein lab. The Spanish HLA lab directors came to Paris every year for the France transplant meeting and also received the new HLA reagents rapidly appearing from pregnancy sera. These laboratories were the embryo of Hispanotransplant. Hispanotransplant was founded in Seville on April 6, 1980. The presidency of the founding was composed of Professor Núñez from Seville, Professor Vives from Barcelona, and myself from Madrid. This was a big success in our fight against Spanish nephrologists, but we were fully supported by the Spanish government at the Kidney Patients Association. In this blurry photograph, you can see Dausset with Hilliard Feinstein, the Secretary of State for Health, Professor Segovia, and my PhD thesis director also, and the head of Kidney Transplant Association, Isabel Entero. All were invited to meeting in the Spanish Kidney Patients Association in Madrid. Isabel Entero, myself, and one government lawyer wrote a very similar law, a draft, a very similar law to the French one, which we passed to the Secretary of State, Professor Segovia. And it was published without modification within 1 month in October 1979. This has been the basis of the Spanish transplant success. In 1991, I introduced Dausset as Doctor Honoris Causa by our University Complutense in Madrid. I compared Dausset with Miguel Servet, the Spanish physician and philosopher who was born in Geneva because of discovering the blood circulation and other ideas. He was in the HLA field one of these pioneers and creative discoverers who had also to suffer humiliations when a very good Italian researcher Ruggero Cepellini ripped off the writing of his work presented in a scientific meeting before he received the Nobel Prize. Finally, I would like to point out an intimate Dausset ceremony. He sometimes dressed at home the academy dressings and ring given by University of Madrid at his honoris causa doctorate. This stresses the impact that had this fact in his life, no doubt linked to his wife, Rosita, from Madrid. Now I will talk about some of the works we have done, pushed and influenced by Dausset and Edgardo Carosella lately. Recently, the HLA region has been shown to contain HLA class 1 E, F, and G loci. G and probably E and F proteins are immunosuppressive at many levels, as shown by Carosella and others. These loci of non-classical class 1 antigens are apparently less polymorphic than the classical ones. Only 14 HLA proteins and 2 null alleles have been found. The HLA-G polymorphism is mostly found in the alpha-1 and the alpha-2 domains. And now there is a hot debate about HLA-G and also E and F proteins' precise function and significance, and a study of these HLA-G molecules throughout primates during the last 60 million years evolution may give an important clue to still non-answered functional questions. This is again to remark that the HLA-G locus is telomeric to HLA-A locus. Here you can see represented 3 generations of a big human family. Several members do not have the normal HLA-G molecule, both females and males. HLA-G immune suppressor molecules are expressed in the fetus in order to avoid a rejection by the mother immune system. However, each member of this family lacking HLA-G full molecule, homozygous for null alleles, gave birth and were born without problems. No immune deficiencies, tumor, or autoimmunity was detected. This suggests that just a part of the molecule may suffice for this function. Chimpanzees show a low HLA-G polymorphism, just 2 proteins. Gorilla also shows a low HLA-G polymorphism, just 1 protein. Orangutans show 5 different proteins, 5 different HLA-G proteins. All middle-sized monkeys from genus Macaca and others, which now live in Africa and Eurasia, have a stop codon in all MHC molecules. They do not have the complete HLA-G molecule expressed. Still, the alpha-1 domain may suffice for HLA-G function. Otherwise, alternative molecules may undertake HLA-G function. However, New World or South American monkeys that separated from African monkeys more than 50 million years ago show an HLA-G molecule with a lot of variability. This polymorphism is typical of a classical class 1 protein, and in these monkeys, almost certainly by function, almost certainly they function as classical peptide presenters. This as you see, these are the many polymorphic sites that these small South American monkeys have. This is a tree showing the time of appearance in million years ago, appearance of primates on Earth. 50 million years ago or before appeared what now are New World monkeys. New World monkeys. South American started separating Africa sometime about 60 million years ago. MHC-G genes have been suffering different changes, which are, I'm not going to detail them, but they have been suffering different changes over the primate evolution. For instance, no medium-sized African Eurasian monkeys like macaca, as I have said, all of these, they don't show a complete HLA-G molecule. While some humans do not have the complete HLA-G protein, neither. It seems that the immunosuppressive HLA-G molecule is important for fetus acceptance and other immunosuppressive functions in every species, but every species has its own features. The other topic related to major histocompatibility complex and the evolution of the major histocompatibility complex is the evolution in wild small songbirds. Birds consist of about 10,000 species. Songbirds or small birds are about half of them, 5,000 species. Now, this is the wild chicken from Indochina forest. We must remember that the major histocompatibility complex was discovered in chicken and not in mice. If we have 2 billion living chicken today, now, they will have disappeared in human mouths within a month. This means that we have been studying a very inbred chicken population that may have changed. They may have changed the major histocompatibility complex in the sense of that chromatids are quite similar, and DNA exchange between these chromatids are enhanced. This would be probably the cause that the introns in chicken MHC, are so small, 300 bases. And the high polymorphism also found in the chicken. I have shown already the chicken major histocompatibility complex. I'm not going to get in detail into this. In fact, MHC introns in chickens are very short, and this is because this major histocompatibility complex has been called minimal essential MHC. We should name artificial MHC. What happened is that from a very few wild chickens from Indochina forests, we have obtained many brother-sister breedings now, as Takahashi Gohobori demonstrated about 8 years ago. Both a similar degree of inbreeding may be found in our other 2 usual models for major histocompatibility complex studies, the laboratory mouse and humans. This is because we think that this high degree of variability found in MHC is artificially induced by 2 many homologous sister chromatids, and decided to start studying wild small sunbirds. This incidentally, I would like to tell you that you see here a red canary. The canary is living in the Atlantic island and is green, of course. But you can see here who made the red canary in 1930. In Bremen, in 1930, these two associates introduced the red plumage genes of Venezuelan Carduelis cuculata into a canary, into a wild canary, in the European, which lives in the European Atlantic island, as I told you. This was the first genetically engineered animals in 1930. Genetically engineered animals. It was got by back crosses, but it was genetically engineered because they were looking for introducing genes into one animal by one way or the other. So we have studied many wild sunbird species, and we are going to refer to a family of 3 species from genus Carduelis, seaskins and goldfinches. Wild animals have been, wild vertebrates have been studied, but just in single species, but not genera. So looking at the genera with a group of these small bird species, we are able to get more conclusions about the MHC evolution. This Eurasian seaskin appeared 5 million years ago. He has 2 genes, 20 alleles, and 8 proteins in the MHC class 1 locus. It is the evolutive father of the American seaskin, which is the Pinus siskin, which appeared 2 million years later, 3 million years ago. It has 2 class 1 genes, 8 alleles, and 8 proteins. In addition, his relative, the black seaskin, which appeared on Earth 1 million years ago only, had and it is endemic of on the Titicaca Lake region in the Altiplano between Bolivia and Peru. Has also 2 genes, 12 alleles, and 8 proteins. In addition, these songbirds do not conserve these residues marked here on the molecule, which otherwise are maintained from reptiles to humans in all species studied so far. This might support the hypothesis that these small birds are the closest descendants of dinosaurs, as many people put forward. Also, these 3 shows, you see here the same allele from different species, the same allele from different species, 4 alleles from different species. Also, we can see that the same MHC class 1 protein, also these 3 shows that several wild sunbird species share the same alleles, pointing out that there is little natural pressure for diversification. In other words, a strong trans-specific evolution of vertebrates can deal with microbes with a relatively short MHC variability. Also, we can see that the same MHC class 1 protein allele was present in the Pyrenees European seaskins 5 million years ago. Look at number 2, at yellow number 2. And this yellow number 2 is found 3 million years later in one of its relatives, which is the American siskin or the Pinus siskin. Also, the allele named the 4, as you see here, and circle is in red. You can see that it was present in the Pyrenees in the present 5 million years ago, and it is also present in these species, the black siskin, which only appeared on Earth 1 million years ago. In conclusion, this model of wild vertebrates genera do not support the existence of a natural, very polymorphic major histocompatibility complex, as we see in the what we call artificial human, mouse, and chicken models. All have undergone through a clear bottleneck, and also a big inbreeding because of a rapid expansion. This also confirms isolated observation of major histocompatibility complex low variability in certain wild species, not in general, like cheetah or Syrian hamster. So thank you very much. Thank you very much. This is all.